back to Edutainment with Adi. In this video, you have 12 statement type questions in molecular biology. Let's start. Question number 1. Consider the following statements regarding lac operon regulation. Statements are 1. Allolactose act as true inducer of lac operon. 2. Catabolite activator protein CAP enhances transcription in presence of high glucose. 3. Lac operon exhibits both positive and negative regulation. So here the correct answer is option B, 1 and 3 only. That is, allolactose act as true inducer of lac operon and lac operon exhibit both positive and negative regulation. Here the CAP enhances transcription in presence of high glucose. This is the wrong statement. CAP enhances transcription in presence of low glucose. Next question. Which of the following statements are correct regarding catabolite repression? Option statements 1. High glucose concentration reduces cyclic AMP levels. 2. CAP-CAMP, that is CAP cyclic AMP complex, promote efficient transcription of lac operon. 3. Catabolite repression enhances lactose metabolism in presence of glucose. 1. High glucose concentration reduces cyclic AMP levels. That is correct. 2. Cyclic AMP CAP complex promote efficient transcription of lac operon. That's also correct. 3. Catabolite repression enhances lactose metabolism in presence of glucose. That is wrong. So the answer is option A, 1 and 2 only. Okay. Next question. Consider the following statements regarding operon concept. 1. Structural genes in an operon are transcribed as polycystonic mRNA. 2. TRP operon is a repressible operon. 3. Repressible operons are generally involved in catabolic pathways. So, structural genes in an operon are transcribed as polycystonic mRNA. That's correct. TRP operon is a repressible operon. That's also correct. Repressible operons are generally involved in catabolic pathways. That's wrong. So, here the correct answer is 1 and 2 only. Option A. Next question. Fourth question. Consider the following statements regarding DNA polymerases in prokaryotes. DNA polymerases in prokaryote. Statement 1. DNA polymerase 1 possesses 5' 3' exonuclease activity. 2. DNA polymerase 3 is the major replicative enzyme. 3. DNA polymerase 2 synthesizes RNA primers. So the statement is regarding DNA polymerases in prokaryotes. First statement, DNA polymerase 1 possesses 5' 3' exonuclease activity. That is correct. Second, DNA polymerase 3 is the major replicative enzyme. That is also correct. DNA polymerase 2 synthesizes RNA primers. That is a wrong statement. So here the correct answer is option A, 1 and 2 only. Option A, 1 and 2 only. That is, DNA polymerase 1 possesses 5' 3' exonuclease activity and DNA polymerase 3 is the major replicative enzyme. Then next question. Which statements are correct regarding telomeres and telomerase? Which statements are correct regarding telomeres and telomerase? 1. Telomerase is a ribonucleoprotein enzyme. 2. Telomerase contains an RNA template. 3. Telomerase activity is absent in germ cells and cancer cells. First statement, telomerase is a ribonucleoprotein enzyme. That is correct. 2. Telomerase contains an RNA template. That's also correct. And third statement, telomerase activity is absent in germ cell and cancer cell is incorrect. So here the correct statements are 1 and 2 only. Option A is correct. Sixth question. Consider the following statements regarding replication fidelity. Consider the following statements regarding replication fidelity. 1. Proofreading activity reduces mutation rate. 2. 3', 5' exonuclease activity is associated with the proofreading. 3. DNA ligase perform proofreading during replication. Proofreading activity reduces mutation rate. That is correct. 3' 5' exonuclease activity is associated with the proofreading. That's also correct. And the third statement is incorrect. DNA ligase perform proofreading during replication is incorrect statement. So here the answer is option A, 1 and 2 only. Seventh question. Which statements are correct regarding eukaryotic DNA replication? Eukaryotic DNA replication. Statements are multiple origins of replications are present. Okazaki fragments are shorter than in prokaryotes. DNA replication occurs throughout the cell cycle. So first statement, multiple origin of replication is present in eukaryote. That's correct. Okazaki fragments are shorter than in prokaryote. That's also correct. Third statement is incorrect. DNA replication occurs throughout the cell cycle is not correct. So answer is option A, 1 and 2 only. Eighth question. Consider the following statement regarding translation. 1. Translation occurs in ribosome. 2. Stop codons code for amino acids. 3. mRNA act as template during protein synthesis. Statement 1. Translation occurs in ribosome. Is true. Stop codon code for amino acid. It's a wrong statement. Stop codon doesn't code for any amino acids. mRNA act as template during protein synthesis. That is also true. mRNA act as a template for protein synthesis. So here 1 and 3 is correct. Answer option A, 1 and 3 only. Ninth question. Consider the following statements regarding point mutations. 1. Transition mutation involves purine to purine substitution. 2. Transversion involves purine to pyrimidine substitution. 3. Sickle cell anemia results from frame shift mutation. So, the question is about point mutation. First statement, transition mutation involved purine to purine substitution. That is correct. Second, transversion involved purine to pyrimidine substitution. That's also correct. Sickle cell anemia results from frame shift mutation. That is wrong statement. Sickle cell anemia results from point mutation. So, here the answer is option A, 1 and 2 only. 10th question. Consider the following statements regarding RNA polymerases in eukaryotes. 1. RNA polymerase 1 synthesizes rRNA. 2. RNA polymerase 2 synthesizes mRNA. 3. RNA polymerase 3 synthesizes tRNA. So, the given statements are regarding RNA polymerases in eukaryotes. First statement, RNA polymerase 1 synthesizes rRNA. 2. RNA polymerase 2 synthesizes mRNA. 3. RNA polymerase 3 synthesizes tRNA. Here all the three statements are correct and the correct option is option D, 1, 2 and 3. Question. Which statements are correct regarding promoters in eukaryotes? Which statements are correct regarding promoters in eukaryotes? 1. Data box is located upstream of transcription start site. That's correct. 2. Cat box functions as regulatory sequence. That's also correct. 3. Promoters are recognized directly by ribosomes. Promoters are recognized directly by ribosomes. That's incorrect statement. So 1 and 2 is only correct and our correct option is option A, 1 and 2 only. 12th question. Consider the following statements regarding hnRNA processing. 1. 5' capping protect mRNA from degradation. 2. Polyadenylation occurs at 3' end. 3. Splicing removes exons selectively. Consider the following statement regarding hnRNA processing. 1. 5' capping protect mRNA from degradation. That's correct. Polyadenylation occurs at 3' end. That's also correct. Splicing removes exons selectively. That's incorrect. Splicing removes introns. Okay, correct answer is option A, 1 and 2 only. So that was our last question. Hope you understood. See you in next video.